I would like to invite our speakers and panelists to come forward. So in addition to the speakers we've heard from, we have panelist Todd Skaar, who's a professor of medicine and clinical pharmacology at Indiana University, his expertise also with pharmacogenomics, and Serena Jingchuan Guo, who is the William S. Bucke Associate Professor in the Department of Pharmacy Practice and AI Director for the College of Pharmacy at Purdue University and an investigator for Regan Streif Institute. So first I'll ask Todd or Serena if you have any thoughts or comments on what you've heard or any additional information on the talks so far.
Go ahead.
Okay, I'll start. Yes, it was very fascinating. The whole morning presentations and panel and yeah, especially the case studies. Because my background is real-world evidence, we've been working in the field for many years. I want to address. We have a discussion about the role of AI and LLM. And like how much it can replace researchers and technicians, data analysts. Actually, because I work specifically in causal inference, and to our experience by now, there's no single really model can give us very solid clinical causal reasoning. And that's something that, I was thinking to address during the panel, and we can discuss more with use cases.
It's not working, yeah. I agree, fascinating morning, lots of cool things. I guess one of the, things I was sitting thinking and taking some notes on is, as the AI, stuff is taking over lots of things, what are the main studies we need to be doing that generate the key information that goes into those AI things, right? The AI stuff, I imagine, is going to evolve over time because there's, all kinds of big, not even non-healthcare places that are going to develop that, but it's only going to be as good as the key data. So, what sort of things can we recommend to NIH as priorities for, these are really the key data that 2, 3, 4 or 5 years are going to be the key things that are going to make that AI accurate instead of, just generating a lot of, or making it more accurate than that was, I guess, one of the sort of things that we should be talking to NIH probably about.
Great. So I think the examples we've seen both from ClinG PGX and from Liang's examples are in looking at safety issues in labeling, right? And I suspect the bar is a little easier to get through when you're dealing with a safety issue. For instance, from a maternal standpoint, the black box warning on using codeine in breastfeeding was added based on a case report that's now been withdrawn. So now the question is, how do we look at removing that black box warning? But also, how do we incorporate things that maybe are more of a therapeutic or a beneficial use from medication versus a safety use? And so, what level of evidence is really needed to inform those decisions?
Yeah, I would say, definitely. I think the safety issues are going to be, bigger on their radar. I didn't go into citizen petition. That was done in the case of fluoropyrimidines, my example. But I think the concerning part is like the number pending and like how long have they been pending. So I think that sometimes, things might not be outright rejected by FDA, but that's what I'm talking about, resource limitations and so forth, them being aware of certain things have been filed and they're aware of these things, but things move rather slowly sometimes. And I think that huge safety issues, like either things that are extremely severe or things that affect large numbers of people are going to be probably more successful. So if you have examples that, like where you can show that kind of information, I think probably will be your best bets.
Great. And I'll note that Terry Klein has made some comments in the chat that Todd's correct, that it's making the data available so that it can get incorporated into foundational and large language models is critical. And then also that based on what Lang's presentation, Sean's experience is that public advocacy is your best bet. So engage in patients and those groups. They are the strongest way to move forward for labeling changes. So we'll turn it to audience questions. Prince?
Yeah, so follow up to that. So I was struck that, 37% of those were still pending. And so I pulled out the paper and they explained that there's no legal requirement for them to respond in any specific time frame, so they could just choose to not respond. So that's for citizen, applications. Is there a mechanism for other professionals to respond where they actually have a set timeline?
Not that I'm aware of, but I'm not a regulatory expert.
Yeah, I can talk about that a little bit. Usually when FDA receive the applications, citizen petition, they will classify that based on whether it will affect pending application or not. If yes, there will be a timeline. You need to resolve that before in consistency with the application review clock. So they will be bundled together. Otherwise, there's no timeline. The people have been abusing the mechanism. I've long mentioned for example, there are so many, the large amount of petitions about to block, to make the regulatory standard higher for generic entry. So, so the drug's so risky, you cannot let another copy go to the market with such less trials, then that's why FDA need to reject most of those.
Does the timeline sometimes restart because you're talking about having a question as well? So if FDA responds to something or asks a petitioner for questions, it kind of sometimes can lengthen the process as well, right? They're asking for more information, then they have to respond, and sometimes they will ask again. Sometimes there's many questions involved.
Yeah, many disciplines.
Yeah, I was going to add on, and Phil. Yeah, and Phil Wilson in the chat is also saying the BPCA provides support for studies directed at providing pediatric label of off-patent drugs and through using their processes and working perhaps with PTN, whether BPCA might be asked to use real-world evidence data collected by MPRINT to relabel benzonatate.
And also, I think, FDA has the MedWatch program, and Dr. Zhao might know that actually that's a spontaneous adverse event report based on data. About 95% actually are from a pharma, because that's mandatory, and 5% came from providers and individual patients that's volunteer.
Dr. Lee, thanks for a great talk. I really appreciated the methodology used to try and improve drug safety. I had a question. So I'm a pediatric cardiologist, and as a pediatric resident previously, took care of patients who came into the ED with various illnesses, and then we may have diagnosed them actually with febrile seizures, so not a primary seizure disorder, but had a high fever that then caused the seizure. I guess I was curious in the different clinical features that you mentioned you adjusted for in the analysis. You had diagnosis, but I was curious if you adjusted for if the patient did or didn't have a fever. The drug you mentioned, it seems like has a clear mechanism of action to potentially cause seizures as a sodium channel blocker, but I was just curious if that could have confounded the results.
Not necessarily individual disease as indicated being justified in model, rather than using several variables characterize the disease, just say health conditions as covariates, but not necessarily individual disease yet.
Okay, so not necessarily if they had a fever at the time of the seizure.
No. We have not done anything on that scale yet. That's where we need to collaborating with more physicians, make sure the signal we saw is not flu. And then the other thing I want to say, kind of circle back to Serena's comments about causal inference framework. So there's obviously a population-based approach where you can using, controls, right? Try different controls, trying to remove the confounding as much as possible. Maybe either in the regression, you're using more cutting-edge causal inference technology to do it. That's one approach to do it. The other one is a case report. So I think in the medical domain, there's also a big trend on the case report analysis. They call it causality assessment on the case report too, and people using that as well. So we probably going to do both, strong evidence from case report and also population-based approach and to see whether they're consistent. So still a lot of work has to be done, put this way. So we're definitely going to do our best, trying to distill all the signals and trying to figure that out. So I'm glad you're asking a very clinical relevant question.
Thanks so much.
Okay, follow up on that. Do you have any, with the clinical data that you were adjusting for, do you have any way to sort out to make sure that the reason the patients got that maybe was because some of the other drugs, I think like codeine and dextromethorphan, are seizure risk? So it might be that the patients that got that were the ones that have, for some reason, an enhanced risk of getting seizure, and you're just seeing that all the patients were shifted over to that from instead of the other ones?
Yeah, medication, co-medication. We have not done that much yet.
Hi, thank you all for a really interesting session. So my question also relates to the label change topic and maybe introduces a little different perspective. So, a complementary approach to relying on pharmacovigilance and post-market data for updating labels is to gather more patient-reported outcomes during clinical trials. So in that sense, the labels may not need to be updated because they've been right all along, or at least more right. So I'm curious, how you think about those two things, gathering PROs versus relying solely on post-market information for updating labels, and how they might together be more than the sum of their parts in that context.
Okay, so when you look at the drug labels, there are, I'm just talking about toxicity, right? So there are very generic description of the symptoms, which sometimes is not very much helpful for either physician or patient. And I think it's all about the severe symptom. I would say, put it this way, when you look at FDA drug label, that symptom related to the drug is not necessarily, they say causal, say highly important, like box warning and severe ADEs. It's not really say causal, it's really about, kind of evidence strong and strong association. That's how it is. So what I think the impact we're trying to make though is really about whether if we find that benzonatate has strong possibility of associated with seizure or arrhythmia, we want that things being put in a more highlighted session like box warning or severe ADE, that session. So that's where the goal is. But all the clinical trial data being collected from AE data, typically they're presented in the label, but not necessarily kind of in a highlighted session, right? So that's the difference.
I actually do have another unrelated question if nobody else has one. So it stems from Dr. Zhao's talk, although the answer doesn't have to be limited to him. But I'm curious, you sort of painted this vision at the end for an end-to-end, AI-powered drug discovery and trialing workflow. And I'm curious, how amenable our current regulatory environment is to each phase of that, and what any of you might see as the biggest regulatory changes that would need to happen to be more amenable.
It's a great question. Thank you for asking that. It's also related to the labeling question. I think labeling, so for example, if you're using post-marketing data in real-world evidence, in FDA, making a change in labeling is a big impact. Usually the observational studies are not considered as strong enough unless it's really, really life-threatening. Usually people conduct like additional clinical studies to change the label. So any real-world evidence making a labeling impact, I don't know how many number of that, my guess should be rare. So coming back to the end-to-end tools is still something that the industry, entertaining, not by FDA. FDA, just as I mentioned earlier, just want to review the pivotal trial data. Is evidence-based agency. So if in the predetermined trial, pivotal trial, the data, efficacy, safety, matched to the predetermined preset standard, like a statistical analysis plan ahead of time, then the drug will be approved. In my opinion, there could be revolutions in the future, especially given the current commissioner. They are talking about using plausible mechanisms, using like for super ultra rare disease, and also they are talking about rare disease evidence principles. Those things you can leverage sometimes the preclinical data, and then even some in vitro data or binding data or animal data, but those things are rare. End-to-end model is meant to increase industry's revolutionary power to short the development timeline, but you still need to generate the confirmatory evidence for FDA to review. FDA is safeguarding human health. You cannot just say, invite a Nobel Prize winner on the table, he believes it works, then it will work. We've seen in the industry meetings, I've seen so many people, such dynamics. This famous person being invited on the table, even in AC meeting, but I believe this mechanism is going to work. The FDA, even I, as a small guy, can't tell a Nobel Prize winner. So FDA is an evidence-based agency, so show us the data. So the end-to-end model is mainly for tech and farmers to adopt, to make the process faster, more to also to increase the success rate of drug development. That's so far where it stands.
Can I ask sort of a related question that I think with your FDA experience, as we're generating post-market data that we think might influence labeling and change that. So in contrast to all the drug discovery and the initial studies done for approval, which are all done in very expensive labs with CLIA, purely CLIA conditions and all that stuff. A lot of the studies that we do that are funded through NIH and other places, the genetic stuff may not be done in a CLIA tested or a CLIA validated lab. They're done in research level labs. Same with the drug analysis type stuff. So what sort of things should we be thinking about, or I guess another way to say it is, as we're doing these things, when does that stuff need to be done in a CLIA, under pure CLIA conditions equivalent to a drug discovery, pre-drug approval, or when is it okay to do it just under good clinical practices, but don't have all the CLIA validation, where you mark down every time you leave and get a drink of water and whatever, versus what is okay just as our typical research lab stuff.
Yeah. I'll add to that not even CLIA certified, but GLP or GXP certified labs or FSMA compliant databases. So. Yeah,
I think for GLP, right?
Yeah, where we don't have that.
You don't have that. I know for Pharmacotherapy study, you need later on in the exploration stage, you can do anything, but in the really the first time in human study, the data to support that, you need to be in the GLP lab. You need to follow those kind of requirements. I'm not a CMC reviewer, but that's part I know. For the real-world evidence, like pharmacovigilance, I think FDA has the office OSIS. They has been really trying hard to get into the core drug review team. There will be real-world evidence in combination with the other evidence in the package. It's mainly used as a supportive evidence, I will say. It may be changing, but I think the new commissioner is advocating some new initiatives. FDA, one thing is real-world evidence. That's after I left the agency. I left the agency 4 months before Trump took the office. So after that, there are some changes like real-world evidence and AI initiatives. I think the paradigm may be shifting, but that was status quo when I was leaving the agency.
All right. I don't see any other questions online or in the room. So with that, I'll thank everyone for an interesting session. It looks like we have a lot of work to do. And as Erin has pointed out, as MPRINT, we are able to submit requests through the 409A mechanisms developed by the BPCA, at least for the pediatric component of labeling. So I suspect we will be looking at doing work in that area in the future. And as we have been hearing since the beginning of MPRINT, bringing in patient and community advocacy groups is a critical step as well. So Liang, do you have a closing thought?
No, we were looking for collaborations.
Yeah, and that's an example of where having the scientific and clinical expertise with the bioinformatics team is critical to make sure we're looking at the right confounding factors as well. So, and it makes the group stronger. So with that, I will close our meeting today.